Good morning, it's February 6th, and this is your daily brief in science. Here's everything you need to know. A new frontier in cancer treatment may be emerging from the Dana-Farber Cancer Institute, where researchers have reported promising results from a clinical trial involving a personalized cancer vaccine. This trial focused on patients with stage 3 or 4 clear cell renal cell carcinoma, and notably, all 9 participants generated an anti-cancer immune response. The vaccine was specifically designed to target neoantigens, leading to a remarkable 166-fold increase in vaccine-induced T cells that persisted for up to 3 years. This indicates a robust immune response that could be crucial in the fight against cancer. Administered post-surgery, the vaccine aimed to help patients' immune systems recognize and eliminate any remaining tumor cells, and all participants remained cancer-free at a median follow-up of approximately 34.7 months. In this trial, 5 of the 9 patients also received ipilimumab alongside the vaccine, which enhanced the overall treatment approach. The findings suggest that neoantigen-targeting vaccines are highly immunogenic and can effectively target key mutations in kidney cancer, leading to significant immune responses. Despite these encouraging results, experts stress the importance of conducting larger randomized trials to confirm these findings and address limitations such as the small sample size. Phase 1 trials like this primarily assess safety and tolerance, and further research with larger groups will be necessary to determine the long-term efficacy of these vaccines. An ongoing international randomized study is set to explore the combination of the neoantigen-targeting vaccine with pembrolizumab for patients at high risk of recurrence after surgery. The vaccines were customized to recognize the individual cancer of each patient, utilizing tumor tissue to extract molecular characteristics that differentiate tumor cells from normal cells. With a favorable toxicity profile, this vaccine shows potential as a monotherapy or in combination with immune checkpoint inhibitors in treating renal cell carcinoma. The research was led by David A. Braun and Tony K. Choueiri, with findings published in Nature and supported by the Gateway for Cancer Research and the Department of Defense's Kidney Cancer Research Program. While existing cancer vaccines have largely yielded modest results, new approaches like neoantigen vaccines offer hope for more effective treatments. Scientists at Oxford University have reached a significant milestone in quantum computing by demonstrating the first instance of distributed quantum computing. This breakthrough connects 2 quantum processors through a photonic network interface, marking a pivotal moment in the field. The innovative architecture employs modules containing trapped ion qubits that are interconnected via optical fibers. This setup allows quantum logic operations to be performed across these modules through a process known as quantum teleportation. By utilizing photonic links, the researchers successfully executed quantum teleportation of logical gates, effectively wiring the processors together for enhanced computational capabilities. Their findings, published on February 5, 2025, in the journal Nature, indicate that network-distributed quantum processing is feasible with current technology, although scaling up remains a significant challenge. The researchers validated their approach by executing Grover's search algorithm, showcasing that distributed quantum computing can outperform classical computers in searching large datasets. This breakthrough addresses the long-standing scalability issue of quantum computing by enabling small quantum devices to be interconnected rather than relying on a massive single-device setup. Lead researcher Dougal Main emphasized that their technique facilitates interactions between distant quantum systems, effectively connecting separate quantum processors into a cohesive computing unit. This study marks the first successful demonstration of quantum teleportation of logical gates across a network link, potentially paving the way for a future quantum internet that could enable ultra-secure communication and computation. Professor David Lucas, the principal investigator, noted that while the experiment demonstrates feasibility with current technology, substantial engineering and new physics insights will be necessary to scale quantum computers effectively. The modular approach mirrors traditional supercomputers, which enhance capabilities by linking smaller computers and addressing the engineering challenges associated with increasing qubit density in a single device. A groundbreaking study from the University of Maryland has unveiled new insights into how double-stranded RNA, or dsRNA, enters cells. Researchers used microscopic roundworms as a model to examine this process, leading to the identification of a protein named SID-1, which plays a crucial role in regulating dsRNA transfer. Interestingly, when SID-1 was removed, the worms demonstrated an enhanced ability to pass on gene expression changes to their offspring. This finding suggests that understanding SID-1's function could lead to targeted treatments for human diseases and improved control over disease inheritance. In addition to SID-1, the study also discovered a gene called SDG-1, which regulates jumping genes, DNA sequences that can relocate within the genome, potentially affecting genetic stability. Senior author Anthony Jose described the balance maintained by cellular mechanisms as akin to a thermostat, ensuring necessary flexibility while preventing harmful excesses. Jose emphasized that these findings challenge previous assumptions about RNA transport and underscore the role of RNA in inheritance. The research identified multiple pathways for dsRNA to enter worm cells, which could enhance drug delivery methods for human treatments. RNA-based medicines, including RNA vaccines and dsRNA therapies, offer promise in fighting human diseases, but often struggle with efficient cellular delivery. Notably, changes in gene expression persisted for over 100 generations, even after SID-1 was restored, indicating a significant impact on inheritance. These findings provide valuable insights into gene regulation across generations, potentially paving the way for innovative treatments for heritable diseases in humans. The research received support from the National Institutes of Health and the U.S. National Science Foundation, with future studies planned to explore the transport mechanisms of different dsRNA types and the regulation of specific genes across generations. Researchers at Columbia University have made a significant discovery regarding how the brain controls eating behavior. They identified specific neurons in the brainstem of mice that signal when the animals have had enough food. This research utilized advanced techniques, including spatially resolved molecular profiling, to explore the functions of these previously unrecognized neurons, which are similar to those involved in appetite regulation. The study found that these neurons are activated by glucagon-like peptide 1, or GLP-1 agonists, such as semaglutide, commonly used in obesity and diabetes treatments. Conversely, they are inhibited by hormones that increase appetite. Jeff Davies, an expert from Swansea University, emphasized the importance of understanding brain regulation of eating, especially in a world where food is readily accessible. These neurons produce cholecystokinin, a hormone that plays a crucial role in regulating appetite, underscoring their significance in the satiety process. Lead researcher Alexander Nektow noted that while these specialized neurons were previously found in rats, their location in the brainstem suggests similar neurons likely exist in humans. The findings, published in the journal Cell on February 4, 2025, reveal that these neurons detect food through various sensory inputs, including smell, sight, and tactile sensations in the mouth and intestines. When genetically modified to be activated by light, the mice in experiments consumed significantly smaller meals, demonstrating their role in regulating meal size and slowing eating behavior. This groundbreaking research opens new avenues for understanding satiety mechanisms and could lead to innovative therapies for obesity by modulating these neurons and combining treatments with GLP-1 drugs. A recent study has drawn attention to a troubling correlation between cannabis legalization in Canada and a sharp increase in schizophrenia cases linked to cannabis use disorder, particularly in Ontario. The research reveals that the population attributable risk fraction for cannabis use disorder related to schizophrenia rose significantly from 3.7% before legalization to 10.3% during the legalization phase. It is estimated that around 10% of new schizophrenia cases during this period could have been prevented if individuals with severe cannabis use disorder had ceased their use. This figure is notably higher among young men aged 14 to 24, where the preventable cases increased to 18%. Jody Gilman from Massachusetts General Hospital has stressed the importance of comprehensive epidemiological studies to assess the long-term mental health impacts of cannabis legalization, especially for vulnerable populations. The study utilized extensive health administrative data to analyze the effects of changes in cannabis policy on mental health outcomes over time. The findings underscore a significant public health challenge, particularly due to the increased potency and frequency of cannabis use among youth. While the study does not definitively link heavy cannabis use to the onset of schizophrenia, it suggests that regular users may experience earlier onset and more severe symptoms. Published online on February 4, 2025 in JAMA Network Open, the study was supported by the Canadian Institutes of Health Research and ICES. Investigator Daniel Myron cautions that legalization does not eliminate the risks associated with cannabis, similar to the dangers posed by legal substances like alcohol and tobacco. The notable rise in schizophrenia cases related to cannabis use disorder indicates an urgent need for targeted prevention strategies, particularly focused on younger populations. Following the legalization of non-medical cannabis in 2018, emergency department visits and hospitalizations for cannabis use disorder surged by 270%, highlighting a growing public health concern. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.